Good morning. This is the first part of the molecular basis of inheritance. Why do we call this unit the molecular basis of inheritance? In the previous unit, we studied the inheritance of characteristics from parents to offspring, and today we are going to study the molecular basis of this inheritance. What does this mean? This means that all processes of passage of characteristics from parents to offspring are based on a molecule, and that molecule is extremely important, and it is mainly the DNA molecule. Of course, there are other molecules that share in this process of inheritance and that are crucial for it. For example, RNA, as well as other proteins and other factors that are responsible for inheritance. And this proves that a single molecule like DNA can control many characteristics and can affect inheritance. We will be splitting this unit of the molecular basis of inheritance into 3 parts. Each part is around 60 or 65 slides. And today we are going to discuss the introduction, or part 1. Of course, today we all know that DNA is responsible for inheritance, and it is the molecule of heredity, as many people like to call it. As we said, the molecule is very important as it the sequence of nitrogenous bases on DNA controls, or the sequence of nucleotides on DNA are the genes. So every chromosome is nothing but a DNA molecule. Every chromosome is nothing but one DNA molecule. So one chromosome is equal to a highly folded DNA molecule, one highly folded DNA molecule. And of course, you know that this DNA molecule is highly folded because it has been folded on nuclear proteins, as we have studied before, and those nuclear proteins are mainly histones and non-histones. And you also know that this single DNA molecule, together with the histones and non-histones, form together the chromatin, okay, or a substance called chromatin. If we look at a chromosome, if we look at a diagram of a chromosome, we will find that the chromosome is divided into regions, and every region is a sequence of nucleotide on DNA, as we will see later on. So every chromosome consists of many genes. For example, gene number 1, gene number 2, gene number 3, 4, 5, etc. Of course, the chromosome contains a much bigger number of genes. And in between those genes or those genes or alleles are nothing but a sequence of nucleotides. So they constitute a part of the DNA molecule. A gene is a sequence of nucleotide. A gene is a sequence of nucleotide on a DNA molecule. But it's not just a sequence of nucleotide on a DNA molecule, because in order for it to be called a gene or an allele, it has to be controlling a certain characteristic. So it has to be coding. And by coding, we mean that it codes for a particular trait. And by coding for a particular trait, we mean that this gene will be translated into a certain protein. This gene will be translated into a certain polypeptide or a protein. And through that polypeptide, the gene will control a trait. Why? If we look at the upper part of the screen, because this polypeptide might be, for example, a hormone or a growth factor, or it might be an enzyme, and that enzyme might control a particular chemical reaction. And at the end of that chemical reaction, you obtain a product, and that product is responsible for the trait. For example, the polypeptide you produce through the enzyme might be a pigment. And through formation of hormones and enzymes and pigments, actually, enzymes are the most important polypeptides in the cell, because it is through enzymes and their catalysis of chemical reactions we can produce hormones, we can produce pigments, and therefore pigments and hormones are produced via chemical reactions that are produced or catalyzed through enzymes. Now we can see the location of the genes on the DNA molecule by tagging them with certain dyes that are fluorescent and that allow you to identify the genes. We are not going to study this in detail, but whoever is going to specialize and study more in DNA will know this very well. Until the year 1940, there was a misconception that proteins and not DNA are the molecules of heredity, are the molecules that are responsible for transmission of the characters. It is worth noting that until Mendel's era, the genes were not yet known, because Mendel's rules or Mendel's laws talked only about factors that were transmitted from parents to offspring. So Mendel did not know anything, and this is a bit, maybe it might be a bit weird to you, because we always write genetic diagrams, we always write genotypes and phenotypes, but Mendel just talked about factors. So this is number one. He didn't know anything about the genes, but he knew that every trait is controlled by two factors. So a factor that is dominant and a factor that is recessive. And then later on, the scientists working in genetics or geneticists started to launch the era of molecular basis of inheritance. And they knew a lot about the chemical structure of proteins, and they knew a lot about the chemical structure of DNA. And they knew that DNA consisted of nucleotides, but they didn't know which of them, or actually they were biased towards proteins, thinking that proteins are the molecules of heredity. And they had the reasons to say this, because proteins constitute a major part of the cell, as proteins enter in the composition of the cytosol, as well as in the composition of the organelles. The cytosol is full of protein molecules, partly enzymes, partly hormones, partly pigments, partly transport proteins, and so on. Number two, proteins are very complex and very variable, as you know, because proteins are made of polypeptide chains, and these polypeptide chains vary in length, they vary in the type of amino acids, so they vary in the amino acid types, they vary in the amino acid sequences. And since proteins are that variable and that complex, then they are more likely to be the genetic material. But at that time, they didn't know that this huge complexity and this huge variation in the protein molecule actually arises from the complexity and the variation in the DNA molecule that controls the protein synthesis process. They also thought that proteins are the molecule of inheritance because proteins were highly specific, meaning that, for example, every enzyme controls a specific chemical reaction. And they knew more about proteins than they did about DNA. A side item is DNA staining, because if you look at a tissue, for example, under the microscope, and you find that this tissue, of course, is made of cells, then you will have a nucleus. This is what you can see under the high power, for example, of an ordinary light microscope, if the cells are big enough to be seen under the high power of a light microscope. You will see the boundaries of the cells to a certain extent, but what is more striking in a stained tissue, because if you don't stain the tissue, it would be extremely difficult to visualize the cells. You will see two types of colors, as we said before in human biology. You will see the majority of the cell, or actually the cytosol and organelles, you will see them pink, and you will see that this area of the nucleus is rather blue. Now, this is because when we stain a tissue, we actually put two types of stains together. We put hematoxylin, and this is what you're going to see in your practical work if you major in a branch that is related to medicine or to pharmacy. You stain the tissues with hematoxylin and eosine. They're always treated as if they were twins. We always say hematoxylin and eosine. Why are we putting both together? Because there are certain structures in the cell that will be stained with hematoxylin, and other structures that will be stained by eosine. Now, eosine is a pink stain, so any structure that is stained by eosine, that loves to be stained by eosine, that has high affinity to eosine, will be pink. And structures which have a high affinity for hematoxylin will be purplish blue. Purplish blue. Actually, they are more purple than blue. Okay? Affinity means to love or to attract. Okay? Now, hematoxylin is a basic stain. That's why all structures that will be stained by hematoxylin, like the nucleus, like the DNA molecule, actually the nucleus loves hematoxylin because it is DNA that loves hematoxylin. We call them basophilic structures, and whatever structure that will be stained pink will be an acidophilic structure. Acidophilic structure. Of course, DNA or chromatin, that consists of mainly DNA and some nuclear proteins, is present in the nucleus, appears blue in the nucleus because in staining, because it loves the hematoxylin stain. Now, there are scientists that questioned the fact that DNA is not the genetic material and that proteins are the genetic material. And we are going now to have like a historical review of the efforts of scientists to discover that DNA is the genetic material. Actually, during our historical review, we're going to show how scientists had evidence that DNA is the genetic material. The first experiment, of course, all the pieces of information that were considered evidence for DNA being the genetic material were derived from genetic experiments. But the first and very famous genetic experiment, that's called Griffith experiment, in 1927 was not done for that purpose of proving that DNA is the genetic material, but it was done for another purpose. Griffith was studying pneumonia, and pneumonia, as you know, is inflammation of the tissues of the lung. So pneumonia is inflammation of the tissues of the lung. And pneumonia is caused by a diplococcus. You know, of course, that cocci are spherical bacteria. Diplococci are spherical bacteria that exist in pairs and that can be inhaled and can affect the lung tissue and cause pneumonia or inflammation of the lung tissue. Some of these strains were virulent. And they were capable of causing pneumonia not only in human beings, but also in animals like mice. And other were non-virulent. The main difference between a virulent bacterium and a non-virulent bacterium is that virulent bacteria cause a severe disease. So a virulent bacterium causes, or a virulent strain, causes a severe disease, and that disease might lead to death. Whereas non-virulent bacteria just cause a mild form of pneumonia that doesn't cause death. Griffith experiment showed that these virulent bacteria that cause death contain a particular substance. If this is the bacterium, that is a virulent bacterium. Actually, it is a spherical one because it is a coccus. So if this is the virulent bacterium and this is the DNA, Griffith discovered that these virulent bacteria, when they are cultured together with non-virulent bacteria, could transform them into virulent ones. So scientists, or coming after Griffith, wanted to know what thing was transferred between the virulent bacteria to the non-virulent bacteria to transform it into a virulent one. Actually, that substance that was at that time of unknown origin was called a transformation factor. Whenever we don't know the nature of the substance, we call it a factor, just like Mendel called the alleles factors. Griffith called this substance a transformation factor. So what is the transformation factor? It is a substance at the time of Griffith whose nature was unknown at the time of Griffith, and that substance was transmitted from the virulent bacteria to the non-virulent ones, transforming them into virulent ones. Of course, if I say this to you now, you automatically think that this should be the DNA, because it is the DNA that is capable of controlling the characteristics of an individual. But at that time, they didn't yet know that DNA was the genetic material. The whole process was called bacterial transformation. Okay, so here we have the experiment. We have the virulent strain that we call the S strain. And when we inject the virulent strain into the mouse dies, especially, of course, that this S strain is living. But the other strain, that is a non-virulent strain, is called an R strain. And that strain is non-virulent, as you see, the blue one. And when we inject in the mouse does not die. They say that in the diagram, they say that the mouse is healthy. Actually, the mouse is not healthy. The mouse acquires a mild disease, okay? And then we start heating the culture where the virulent bacteria are present. This is the flame. So we kill them. And now the S strain is called a heat-killed S strain. Why are we specifying? Because there are several ways of killing the bacteria, and this is one way of killing them. And then when we try to inject another mouse with the heat-killed S virulent bacteria, the mouse stays healthy, the mouse does not die, and we cannot find the virus in the heart muscles of the mouse. And this is the essential cause of death, the cause that, or the reason why the first mouse died is that pneumonia was very severe and it reached the heart and caused infection of the tissues around the heart. And now we do something interesting, that we mix the dead virulent bacteria with the live non-virulent bacteria. And we discovered that the mouse dies and that the virus is present in the myocardium of the mouse, which proves that although these virulent bacteria are dead, yet they were capable of converting the non-virulent bacteria into virulent ones, and therefore transformation occurred. Which type of strains showed the transformation? It was the non-virulent strain. So the non-virulent strain has been transformed into a virulent strain. The non-virulent strain had been transformed into a virulent strain. Now, the second experiment that has been done is, I'll pause for a minute. The second experiment that has been done to prove that DNA is the genetic material, or actually it was the first experiment, because Griffith's experiment was just like an accidental observation due to another goal of another experiment. So Avery and colleagues wanted to know what the transformation factor is. So they proved that this transformation factor is DNA and not the protein. So Avery and colleagues wanted to know what the agent that carried the genetic characteristics from the virulent bacteria to the non-virulent bacteria and caused them to be transformed into virulent ones. And they discovered chemically that this substance that caused the transformation was DNA and not the protein. And it was the first time that they discovered that the transformation factor, and therefore most probably the genetic material, is DNA and not the protein. But they weren't yet certain that this is the genetic material. So all what they discovered, of course, this was a very important experiment. All what they discovered is that the transformation factor is DNA. The transformation factor is DNA. This is all what they discovered. So there remains a very important question. Is the transformation factor the genetic material? Yes or no? So they were almost certain. Of course, since it caused transformation, then it controlled the characteristics, then most probably it is the genetic material. But they wanted to be more certain that the transformation factor is the genetic material. So they wanted to be more certain that the transformation factor is the genetic material. Now, the third experiment, which is Hershey and Chase, was trying to answer this question, and it was mainly based on the study of bacteriophages and the labeling of bacteriophages with radioactive material. As its name implies, the bacteriophage is a virus that causes bacterial lysis. Phage is eating or eater, but it doesn't actually eat, but it causes bacterial lysis. So a bacteriophage is a virus that causes bacterial lysis. And it causes bacterial lysis with a very interesting way. The bacteriophage is a very specific virus and very unique virus because it has a head, and that head has a protein coat, okay? And inside the protein coat, there is the core. So here we have the coat, and here we have the nucleic acid core. We have the nucleic acid core. So here we have the protein coat, and here we have the nucleic acid core. And then we have a tail, and that tail is made of proteins, and the tail has like appendages. So what happens is that the virus adheres to the bacterium, and then it starts to inject, like a syringe, it starts to inject its DNA inside the bacterium. So now inside the bacterium, we have two types of DNA. We have two types of DNA. We have the bacterial DNA, and we have the viral DNA. The bacterial DNA and the viral DNA. Now, when the virus injects its DNA inside the bacterium, fragmentation of the bacterial DNA occurs. I'm just reminding you of what we studied in grade 10. So there is fragmentation of the DNA of the bacterium, and now the DNA of the bacterium becomes a useless DNA, And the viral DNA starts controlling the machinery of the bacterium to replicate its own DNA and form viruses. And at the end of the day, the bacterium is useless. The bacterium ruptures and releases thousands of molecules of DNA. All this, of course, is important to understand how viruses reproduce through a lytic process. We call this process the lytic process of replication, and we're going to study it at the end of this unit because it is important. But what happened is that the scientists labeled the protein with radioactive sulfur. Radioactive sulfur is sulfur 35, and labeled the phosphorus with radioactive phosphorus, which is phosphorus 32. Why? Because proteins contain sulfur but no phosphorus, and DNA contains phosphorus but no sulfur. So when the bacteriophage injects its DNA, then what we are going to see inside the bacterium is the radioactive DNA. The radioactive DNA is radioactive because of the radioactive phosphorus. So every time the bacteriophage injected its genetic at that time it wasn't yet known that DNA is the genetic material, but it was known that the material that is injected is the genetic material. So we want to know which material will be injected, because the material that is injected controls the replication of the virus, and therefore that material that is injected by the bacteriophage is the genetic material. So what will the bacteriophage inject into the bacterium? Will it inject the protein or will it inject the DNA? How could we know? Without labeling both protein and phosphorus. When they did this, they found that it was the phosphorus that was injected and therefore the DNA. Actually, Hershey and Chase experiment was the crucial experiment to determine that, and the final one to determine that DNA is the genetic material. And then we had efforts of scientists like Rosalind, for example. She was a geneticist, and she worked in the lab of Morris Wilkins, and she was working with X-ray, and maybe that's why she died early, most probably because of a cancer. And she performed a process called X-ray crystallography. She exposed DNA, purified DNA, to X-rays in a certain way, and she got a picture of an X-ray picture of the DNA molecule. And from that picture, she could realize, or she could discover almost perfectly, the structure of DNA, or the overall structure of DNA. And as we know, the most famous model for DNA is the Watson and Crick model. But actually, the Watson and Crick model was mainly based on the work of Rosalind Franklin. And at the time, Watson and Crick got the Nobel Prize for their model of DNA. Morris Wilkins was still alive, and he shared the prize with them. But at that time, unfortunately, Rosalind Franklin had passed away, and the Nobel Prize, is not given posthumously. And unfortunately, there wasn't any kind of appreciation to Rosalind Franklin. Now, this diagram shows you how X-ray crystallography works. We have an X-ray tube, a tube that can generate X-rays. So you have two electrodes, negative and positive, with a certain intensity of electric current and electromagnetic radiation that is highly penetrating, that has a very short wavelength and a very high frequency, which is X-ray. Will arise from the X-ray tube or the X-ray generator. Of course, you need a very high voltage, as you see here, ranging from 10,000 volts to 40,000 volts. This X-ray will pass through a LED screen. As you know, the LED screen prevents the passage of X-rays. That's why in the past, people who worked in the field of X-ray used to stand behind a LED screen in order to protect themselves. So when you have a LED screen that only has a small aperture, an aperture or a hole in the middle, then all the X-ray will try to pass through that hole. Why are we doing this? Because we want to have a very focused X-ray, just like a laser beam. But in a laser beam, it's very easy because all the waves have the same wavelength. So they're all like a very concentrated beam. But X-rays are not laser rays, and therefore we have to focus them. And then we have here purified DNA that has been treated in a certain manner to make it crystalline, meaning crystalline, as you see, meaning that it has a regular arrangement of the atoms inside. And she was just curious to know what would happen if DNA, she was working in this field of X-ray crystallography on other substances, and she was now curious to know, since DNA is the genetic material, then it is something important, and therefore we have to try to study this molecule. So this crystalline solid, it was amazing that it diffracted X-ray again. Now we are trying to focus the X-ray on the crystal, and this occurred. Why are we trying to focus it on the crystal? In order to have all the energy of the X-ray on the crystal, and therefore to visualize the shape of the atoms, or the arrangement of the atoms inside perfectly. And then what happened is that the crystal diffracted or diverged the X-rays, and when the X-rays hit the photographic plate, she obtained this picture of the DNA molecule. It is as if we were taking a picture of the DNA crystal. Of course, if she wasn't a scientist, then she wouldn't understand what this shape on the photographic plate represents. But through this arrangement, she proposed the double helix nature of DNA. She proposed that the DNA molecule is made of two strands, and that those two strands are wound or coiled around each other in a helical manner, and that's why she called it a double helix. All the data that were later on said about DNA were based on mainly on this X-ray diffraction picture of Rosalind Franklin. She didn't only say that the DNA molecule is a double helix, but she also talked about the distance between the two strands in the double helix and how the distance was constant and how the distance was almost the width of 3 nitrogenous bases. And this is what we are going to say later on, because if this is a strand, so for example, if you have an adenine base here, as you know from your previous study of DNA, you will have a complementary base here, which is thymine, and they will be bonded by 2 hydrogen bonds. Actually, adenine is a nitrogenous base that belongs to the purine group of nitrogenous bases, and thymine is a nitrogenous base that belongs to the pyrimidine nitrogen or pyrimidine group of nitrogenous bases. The purines are known, we draw the adenine base like this, but actually this is a schematic or a diagrammatic representation. Actually, purines are made of 2 rings. Okay, so purines are made of 2 rings, and they are nitrogenous bases because they contain nitrogen, whereas the pyrimidine family, to which thymine belongs, consists of only 1 nitrogenous base, and therefore the pyrimidines consist of only 1 nitrogenous base. So the width between the two is actually 3 nitrogenous bases. Okay. And then we have another group of experiments that were led by Chargaff. And Chargaff explained that DNA can be accurately replicated and repaired only because of its double-stranded complementary nature. Double-stranded complementary nature. We all know now that if we have a strand of DNA and that you have here, for example, adenine, guanine, thymine, adenine again, and then adenine again, and so on. And if this is the template strand of DNA, then you can make another strand on top of this template strand, and that other strand would be complementary, meaning that in front of A, you should have thymine, in front of G, you should have cytosine, in front of T, you should have adenine, in front of A, you should have thymine. This is what we call the complementary nature of the nitrogenous bases. The complementary basis of nitrogenous, complementary rule of nitrogenous bases means that you will always have an adenine, you will always have a purine base that is bonded to a pyrimidine base. You cannot have a purine bonded with a purine, and you cannot have a pyrimidine bonded with a pyrimidine, because the purines and the pyrimidines complete each other. This is the only way there could be. What do we mean by complete each other? This is the only mean they could make a chemical bond between them. So whenever you have a purine base like adenine, you should have in front of it a pyrimidine base like thymine. Again, if you have a purine base like guanine, you should have a pyrimidine base like cytosine. So not only do we always have a purine with a pyrimidine bonded to a pyrimidine, but a specific purine that is bonded to a specific pyrimidine. Okay, I'll pause for a minute. And now we are going to discuss the work of Michelson and Stahl in 1958, and they explained the way DNA material was or is actually replicated. And Michelson and Stahl, they assumed that DNA replicates in what we call a semi-conservative method, just like Francis Crick, one of the two scientists who proposed the DNA model, had predicted. What do we mean by a semi-conservative method first? Before the era of Michelson and Stahl, there were three theories to explain the replication of DNA, or there were three suggested models of replication of DNA. The conservative method means that you're going to keep the DNA molecule as it is, meaning that you have two strands, and those two strands have the hydrogen bonds in between the nitrogenous bases. Of course, this is a schematic representation, and a new molecule is formed while keeping the original molecule completely intact. In the dispersive method, it's exactly the opposite. The dispersive method says that we are going to fragment the DNA molecule into small fragments, and every fragment will have, of course, I can't explain something that is incorrect, but actually in the dispersive method, you not only separate the two strands, but you break down into fragments, and actually at the end of the day, you have two molecules of DNA that are made of fragments of the original DNA and the new DNA. In the middle, we have the one that is most accepted, that is the semi-conservative method, in which you separate the two strands. So this is an old strand, and this is a new strand. And at the end of the day, you form a new molecule on top of every sorry, this is not a new one, this is an old one. Okay, so the purple strands are the old strands, and on top of every old strand, you're going to build a new strand. So at the end of the day, you have two molecules of DNA. So you started by one molecule of DNA, and you ended up by having two molecules of DNA. So this is DNA replication, because you multiplied the number of molecules by two. And actually, this was done as proposed in the model by making a new strand on top of the old strand, making a new strand on top of the old strand. So each of the two molecules that resulted, the two DNA molecules that resulted from the replication, each of them is being made of one old strand and one new strand. In order to confirm this semi-conservative method of replication, they did a very interesting experiment. It's a bit difficult to understand, or to imagine, but it is very interesting. The idea is based on what? The idea is based on culturing bacteria in a medium containing radioactive nitrogen, heavy nitrogen, nitrogen 15. For generations and generations and generations, bacteria, of course, will uptake this nitrogen 15 in order to make their own DNA. So all the nitrogenous bases in DNA, after a certain period of time, after a big number of life cycles, will contain nitrogen 15. And of course, nitrogen 15 has more neutrons than the ordinary nitrogen, and that's why it is called heavy nitrogen. So the bacteria will be denser than the bacteria with the ordinary nitrogen. And then these bacteria that have all their DNA made of nitrogenous bases that contain all their nitrogen in the form of nitrogen 15 will be transferred to another medium where they have nitrogen 14, and now the bacteria will be required to replicate in a new medium to uptake the new nitrogen that is nitrogen 14 and not nitrogen 15. Before looking at the diagram of the experiment, I'll open a new screen or open a screen actually and tell you something. Now, if I have a DNA molecule, if I start by a DNA molecule where all the nitrogen, now the blue strands will be those that have the heavy nitrogen, and the ones that have the light nitrogen or the ordinary nitrogen will be labeled red. So if this is the original DNA molecule, and then if we are assuming the semi-conservative replication, then every one of these two strands will form a new strand that is nitrogen 14. Okay, so we will have, now I need the blue color, we will have two molecules, and each molecule will be formed of an N15 strand and an N14 strand. And then if this one replicates and that one replicates, what would happen? Now, this blue one now is forced to take the nitrogen 14 because we don't have anything other than the nitrogen, we don't have any nitrogen other than the nitrogen 14, okay? And the other one will do the same. This one will do the same, but it is originally a nitrogen 14 strand. Similarly here, that one will be like this, and the other one will form a new nitrogen 14 strand on top of the old strand, okay? So at the beginning, all the molecules were containing heavy nitrogen. So if we consider this, the density, of course, this is heavy nitrogen. Let's say that the density here is our reference. The density of the original total amount of heavy nitrogen DNA is our reference, and we will consider it 100%. Then after the first cycle, the density will be half the density. Why? Because we will have two molecules, that's true, but the density of every molecule is half that of the original molecule, because you have one strand of the nitrogen 15 and one strand of the nitrogen 14. So you will have 50% nitrogen 15 and 50% nitrogen 14. So definitely the density will be less. And then after this, we will have the 25% only having nitrogen 15, while 75% are having nitrogen 14. So actually, because I made a small mistake, so actually if we consider this 100%, our reference, 100% density, because it contains 100% nitrogen 15, then after the first cycle, only 50% will be nitrogen 15, and after the second cycle, 25% will be nitrogen 15, and so on. And after this cycle, only 12.5% will be nitrogen 15. Now, if we look at the diagram of Michelson and Stahl experiment, here, as I told you, the bacteria were grown in a nitrogen 15 culture, and all the DNA is initially nitrogen 15 labeled. So in the flask, you have all of it nitrogen 15. And then we will add the nitrogen 15 labeled bacteria to a nitrogen 14 medium and grow them for 4 generations, meaning that we have 4 life cycles, meaning that the bacteria will reproduce 4 times or will divide by binary fission 4 times. Okay. And now, this is the density. Okay, we start by 100% density, meaning that 100% is nitrogen 15. So what we are doing is to see how much percent will be nitrogen 15. Okay, so here, after the first life cycle, so here this is the beginning, okay? After the first life cycle, we have 100% in the middle of the density. Here the diagram is related to what I said, but the percentage refers to something else. Here the percentage refers to what? The percentage refers to how much percent of the bacteria have a particular density. Now here 100% of the bacteria were at that level, which is the level of nitrogen 15. And then in number 1, 100% of the bacteria are at this level, which is the level that is midway between nitrogen 15 and nitrogen 14, because as we saw in the screen that wasn't saved, that the molecule will be half nitrogen 15 and half nitrogen 14. So we have 50% containing nitrogen 15 and 50% containing nitrogen 14. And then after the third cycle, we have only 25% containing nitrogen 15, as we said, and 75% containing nitrogen 14. After the fourth life cycle, as we said on the previous screen, 12.5% will be nitrogen 15 and 87.5 will be nitrogen 14. And as we proceed further with further life cycles, we will find that since the bacteria only have nitrogen 14 around them, then later on we will have a 6% here and a 94% there, and gradually 100% will be nitrogen 14. So this proved that the semi-conservative method is okay and that it is correct. I'll not explain again the X-ray diffraction analysis because I have already explained it when we met the diagram. And here we have the structure of DNA. Of course, this is the 3D structure of DNA as it is seen under the electron microscope. And you can see that we have a helical shape. Here, this is the Watson and Crick model. So this is the proposed, or the model that was proposed by Watson and Crick. And here you can see the 2 strands winding around each other. The distance from here to there is 1 turn. We consider this 1 turn, and its height or length is 3.4 nanometers, whereas the distance between 2 nitrogenous bases is 1/10 of that distance, and it is 0.34 nanometers. Here you can say a pyrimidine base and a purine base. Of course, you all know that between cytosine and guanine, we have 3 hydrogen bonds. Between adenine and thymine, we have 2 hydrogen bonds. And as you know, the hydrogen bonds are weak in order to facilitate replication, because at the beginning of replication, the 2 strands should be apart. Why? In order to expose the nitrogenous bases. As long as the nitrogenous bases are bonded to each other, they are not exposed. And if they are not exposed, they cannot be read by the enzymes of replication. So we separate the 2 strands in order, during replication, of course, in order to expose the nitrogenous bases, to expose the bases. And when you expose the bases, they can be read by the enzymes of replication, okay? And now, of course, you can see a nucleotide, which is the monomer, because the nucleic acids are polymers. So we start by a nucleotide, which is a monomer, and then we have a strand of nucleotides, okay? That is a polymer or a polynucleotide. It is a polynucleotide. So the strand of DNA is a polynucleotide. And then we have 2 strands that are polynucleotides and that form the DNA molecule. So what does the nucleotide consist of? The nucleotide consists of the thymine, for example, the thymine nitrogenous base together with a sugar that is a pentose made of 5 carbon atoms and a phosphate group. I'm sorry, I'll pause for a minute. Hello again. We are going to continue explanation of the structure of DNA. We were just talking about the nucleotide, and the nucleotide consists of a nitrogenous base, whatever the type of this nitrogenous base is, except, of course, as you know, uracil, because uracil is present only in RNA together with a pentose sugar, together with a phosphate group. And on the next slides, we're going to see how they are connected to each other. I'll make sure I'm recording. Okay. And now we move on to the next slide. This is the nucleotide. Of course, here, since you have a double-ringed nucleotide, then it is a double-ringed nitrogenous base, then it is a purine base. You needn't know from the chemical structure. You're not responsible for knowing whether it is like adenine or guanine, but it is a purine, because here you have one ring and there you have another ring, and as you can see, you have the nitrogen atoms that indicate you that this is a nitrogenous base. And then here you have the sugar, and the sugar is in its cyclic form, as you can see. So here we have the first carbon. You don't see the carbon atoms, but they're there. And there you have the second carbon atom, third carbon atom, fourth carbon atom, and then here you have the fifth carbon atom. And actually in the fifth carbon atom, we have a CH2OH group. So in order to join that sugar, which is a pentose sugar, together with the phosphate group, you have to remove OH from here and remove H from there and do a dehydration synthesis. So they are bonded together by dehydration synthesis, and of course they form a covalent bond, and that covalent bond is an ester bond. It is called a phosphodiester bond. And why is it an ester bond? Because we have here an acid, which is a phosphoric acid, and an alcoholic group that is present in the sugar. Actually, this is phosphoric acid. This is important to know, because phosphoric acid is H3PO4, okay? So originally we had three hydrogen atoms, okay? So here there was a hydrogen atom, there was a hydrogen atom. So one hydrogen atom in the in this OH group of the acid, and then here we have another hydrogen atom, and there we have the third hydrogen atom, and then we have 4 phosphorus atoms, and we have the phosphate in the middle. So why is this negatively charged? Because in the solution, in the nucleoplasm, this OH group loses the hydrogen atom, and here it lost the hydrogen atom, but there is no negative charge because it made this phosphodiester bond. Okay, this sugar is, as we said, a pentose sugar, and actually it is a deoxyribose sugar. There are several pentoses, but this pentose is a ribose, and not only a ribose, but in DNA it is a deoxyribose. Why? Because in the original ribose, that can be better seen in RNA, you have an OH group here as usual. And if it were present, we would write it, but it's not. Why? Because the oxygen has been removed. So it's only the hydrogen that remains. And as you see, we do not write the hydrogen atoms unless they belong to a hydroxyl group. So it is a deoxyribose, and from here comes the nomenclature of DNA, because D stands for deoxyribo, N and A stand for nucleic acid, whereas RNA stands for ribonucleic acid. Ribonucleic acid, and the ribose sugar in RNA does not lose the oxygen atom. Now, it is important to know that the phosphate group carries a negative charge because, as we have seen before, this negative charge on the phosphate group of DNA is what makes the histones bind strongly to the phosphate, because some amino acid residues on the histone molecule carry a positive charge. So the presence of the negative charge on the phosphate group is essential for winding of the DNA over the histone molecules and condensation of the chromosome. Furthermore, the DNA molecule has a particular direction. What is this particular direction? If you look at the molecule, at a strand of DNA, then you will have that one strand has a free phosphate group, meaning that there is nothing after it. And here we have at the other end a free hydroxyl group, meaning that it is not linked to anything below. So the end that has a free phosphate group will be called the 5' end, and the end that has the free hydroxyl group will be called the 3' end. The other strand will be the opposite, will be anti-parallel. What do we mean by anti-parallel? Meaning that its orientation will be exactly the opposite. The free hydroxyl group or the 3' end will be up, and the free phosphate group or the 5' end will be down. Okay. We talked about, of course, the number of carbon atoms. We talked about the numbering of the carbon atoms, and we give them the prime number. So you say on the ribose molecule, you say that this is the 1' carbon atom, this is the 2', this is the 3', this is the 4', and the 5th' is a side chain. So why are we putting the prime? In order to differentiate it from the carbon atoms of the nitrogenous base. We don't say prime when we count the number of carbon atoms or the numbers of the carbon atoms on the nitrogenous base. And we talked about the complementary base pairing and the distance between the two rings and the hydrogen bonds and the packing and the histone molecules. And of course, you know that we have, you know, from your structure of the cell, from your study of the structure of the cell, that we have structures that are called nucleosomes, and that every nucleosome is nothing but 8 histone molecules around which we have a piece of DNA that is wrapped, and that this winding of this first preliminary binding of the DNA molecule around the 8 histone molecules helps folding the length of the DNA molecule 10 times, and therefore instead of having, for example, a length of 1,000 units, we will have a length of 100 units. But as you know, this is not enough for the nucleus to be able to accommodate the DNA, and there should be further folding around another type of nuclear proteins called non-histone nuclear proteins in order to have the fully condensed chromosome and chromatin. And this is the way they are coiled around each other. So here you can see a chromosome, and we uncoiled the chromosome in order to see the histone molecules that form the nucleosome. Of course, you have to, whenever the DNA is highly coiled around histones and non-histones, it's difficult for replication to occur, because we said that in order for replication to occur, we should uncoil a part of DNA. We should separate the 2 strands. So you have to first uncoil, and then you have to separate the strands. And when you separate the strands, you expose the bases. So you uncoil, you separate, you expose. Ribonucleic acid is very similar to DNA, because ribonucleic acid is a polynucleotide. Ribonucleic acid is a polynucleotide, meaning that it is a polymer that consists of several nucleotides that are bonded to each other, but it is made of only one strand. There are several structural and functional differences between RNA and DNA. RNA is only made of one strand, although some types of RNA, like transfer RNA, might be double-stranded in certain folds. For example, transfer RNA is folded like this. It looks like a clover leaf. So in some areas of transfer RNA, you can have double, apparently it seems as if it were double-stranded because of the folding. The second difference is that ribonucleic acid never contains thymine, it contains uracil. So uracil is the one that binds with adenine. The third difference is that ribonucleic acid contains ribose and not deoxyribose. All these 3 differences are structural differences, but there is a functional difference, which is that ribonucleic acid can leave the nucleus and can be broken down and recycled, but DNA never leaves the nucleus. The main DNA, I'm talking about the main DNA. I'm not talking about extra nuclear DNA, which is the mitochondrial DNA or the chloroplast DNA. This is how pyrimidine nitrogenous bases look like. They are, as you know, single based or single ringed. I'm sorry, they are single ringed. In DNA, we can find thymine, we can find cytosine, but we cannot find uracil. Uracil is present only in RNA. You needn't know the formula. You needn't be able to detect which is which just by seeing the formula. But once you see a single ring nitrogenous base, you can see that you can say that it is either this or that, but it is definitely a pyrimidine base. Similarly, we have adenine and guanine, and both are present, of course, in both. We can find them in RNA and we can find them in DNA. You needn't know how to differentiate between them but you need to know that they are double-stranded. And now we're going to talk about the replication of DNA. Now, what is replication? Replication means making an exact copy of something. To replicate something is just like photocopying, to make a copy. But here, actually, we are making an exact copy of the molecule as a whole of DNA via splitting the two strands of DNA, separating them apart, and making the complementary strand of each strand, okay? And as we said, replication is semi-conservative, as we said before, meaning that it is based on the fact that we are going to split the two strands apart and build a new strand on each of the old strands. It was predicted by Watson and Crick, but it was proved by Michelson and Stahl. So first of all, as we told you, we have to unfold the two strands. And then when you have an unfolded part, you still have the hydrogen bonds in between. So you have to use an enzyme that is called a helicase enzyme, because it's actually the one that helps unfolding. And at the same time, the helicase walks on the DNA strands, the two DNA strands that are bonded by hydrogen bonds and that look like a ladder. The enzyme walks on the DNA molecule and unzips. I love this verb because it's very accurately describing what helicase enzyme does. It is as if the two strands were zipped together and the helicase enzyme is unzipping them. And here unzipping is an informal word, but it means it breaks down the hydrogen bonds that lie between every two complementary bases on the two strands, and we will start to build a new strand on each of the old strands. Now, in order for replication to start, it doesn't start anywhere on the DNA molecule. It is a very organized process, and it starts at very specific sites on the DNA molecule. And these DNA sites are called the origin of replication, the origins of replication. It's a plural name. Yes, it's a plural word. And this indicates that we have several specific points on the DNA molecule where we are going to start the process of replication. At every, say for example, that I decided that this is an origin of replication. So the helicase enzyme will split the hydrogen bonds here in the middle of the DNA molecule. Yes, not necessarily at the extremes of the DNA molecule. Not only does the helicase enzyme separate the or break the hydrogen bonds to separate the two strands, but the two strands, it moves them apart from each other, and therefore we start to have a structure that looks like a bubble, and that we call a replication bubble. And since the DNA molecule is very long, so we cannot do this in series. We cannot create a replication bubble, replicate DNA in this swollen part of the DNA, and then go to another segment and start replicating it. This would take much time, a huge time, because DNA is very long. And why would we do this if we can have thousands of helicase enzyme molecules, each working at a particular origin of replication? So actually, during replication, we have thousands of origins of replications, thousands of helicase enzymes, and thousands of replication bubbles. And of course, the discovery of the electron microscope and the ability to visualize all these amazing details has helped a lot in knowing how the process of replication occurs. And this this makes it more speedy, the process of replication, because the DNA molecule is a giant molecule that consists of how many base pairs in? Say, for example, I have a base pair like this on the DNA molecule. Say, for example, adenine and thymine, or guanine and cytosine. How many of these do we have? 3 billion base pairs arranged along the length. 3 billion means 3 billions means 3,000 millions. So it's a huge number, and this is understandable because we have thousands of genes, and every gene is made of tens or hundreds of nitrogenous bases. This is the origin of replication, and this is the bubble that has been formed. And here we will stay for a while on this slide to understand the process of replication. Now, this structure that has been done or created, produced by the helicase enzyme, is the replication bubble. And at the extremes, the right side and the left side here, we have two Y-shaped structures that are called the replication forks. So this is the replication fork, this Y-shaped structure, because it's forked like this, and the structure in the middle is the replication bubble. So how does the process of replication occurs? We always, so if we separate the two strands, each strand will be considered a template for the new strand. Why? Because if you have, for example, guanine here, then the enzyme that builds up the new strand knows that it should get a cytosine here. And if you have an adenine here, the enzyme knows that it should get a thymine here. How do we call the enzyme that builds up? How do we call the enzyme that builds up the new strand? The enzyme that builds up the new strand is called DNA polymerase. Of course, it's an extremely important enzyme in the nucleus, and DNA polymerase, being that important, will have very strict rules. But before mentioning the very strict conditions of DNA polymerase, we should say that we will have this brownish new strand that is built on each template strand. After separating the two strands, we have two template strands, and everyone will build a new one on top of it. The first rule that DNA polymerase adopts is that it always builds the new strand in the direction 5' to 3'. What does this mean? It simply means that the new strand is built in that direction. See the head of the arrow, meaning that at the end, we should always have a 3' end, meaning that we should always have a free hydroxyl group. And every time you add a new, the DNA polymerase adds a new nitrogenous base to elongate the new strand. This elongation will occur at the 3' end where you have a free hydroxyl group. This is the first rule. The second rule is that DNA polymerase cannot initiate this process unless there is an RNA primer, an RNA strand that is built on top of the template DNA strand. And this RNA primer acts like a signal that tells the DNA polymerase that this is where you have to start the process of DNA replication. The third rule is that DNA polymerase has to build two strands, one on top of this template strand and one on top of the other strand. But the problem is that if DNA polymerase, as it is walking along the DNA molecule, tries to build the new strands at the same time, it will build this strand in the 5' to 3' direction. It would be okay, it is fine. And why am I saying this? Because when you build a new strand, you have to take care of the direction of the template strand. So if your template strand is 3' to 5', then the new strand will definitely and inevitably build in the direction 5' to 3', because this is how a DNA molecule is. They should be anti-parallel, so there is no choice. Now, imagine now that I'm drawing the whole DNA molecule. If this strand of the DNA molecule is 3' to 5', then the other strand definitely should be 5' to 3'. So imagine that we have this poor DNA polymerase that is trying to satisfy everyone, and that is trying to build, this is the polymerase enzyme, and it is trying to build the other strand at the same time as it is walking in this direction. Then if it does, then it would build the new strand in the direction 3' to 5', which is impossible because DNA polymerase cannot do this. It can just add new nucleotides at the 3' end, and therefore it can only build in the direction 5' to 3'. So this is a cause effect that you have to know. So this is important to know. DNA polymerase can elongate the new DNA strand, can elongate the new DNA strand by adding only is very important here, only by adding new nucleotides to a 3' end. And a 3' end is that end where you're going to have a free phosphate group, free hydroxyl group. Okay, so DNA polymerase can elongate the new strand only by adding new nucleotides to a 3' end. And therefore, according to this, it cannot build the two strands at the same time. It cannot build the two strands at the same time. So what will DNA polymerase do? It will turn, it will make a U-turn and build the new strand in this way. In this way, if it builds the new strand, yes, it will be building in the direction 3', 5', and this is okay because 5' here is complementary to 3' here, and 3' is complementary to 5'. So the other template strand, the other blue template strand, has to wait until the first strand has built up its own complementary strand. And meanwhile, DNA polymerase gets tired. And therefore, if for the first time, it built up the new complementary D strand one shot, but here, when it turns, it makes the U-turn, it will make it in the form of small segments called Okazaki fragments. And every Okazaki fragment, as you see here, for example here, every Okazaki fragment is nothing but the fragment of DNA that is built together with its specific primer, because every primer or every Okazaki fragment has its own primer. Of course, the helicase enzyme can continue here and build this one in, if we start here, okay, if it tries to build it, so it will build this new one shot, and then when it makes a U-turn, it will build it in Okazaki fragments. Okay, so we have almost explained all the information here, And here again, we have the same, but we have more details. For example, this is the part of the DNA that has been unwounded, and then this is the DNA primase that builds the RNA primer. And then we have the Okazaki fragments. And there is here a DNA ligase that helps joining the Okazaki fragments together. The strand that has been replicated in one shot is called the leading strand, whereas the other strand that is made up in segments is called the lagging strand. The leading strand is so called because it is the leader. It is the one that has been replicated first in one shot. The lagging strand is lagging behind. It has to wait for the leading strand to be replicated, and it is replicated in Okazaki fragments. And this is the helicase enzyme, and this indicates you that we have separated the DNA in this direction, the two DNA strands in this direction, and the DNA polymerase now forms the new strand in this direction and forms the lagging strand in the other direction. Okay, so we have explained all this, and we have explained the directions. We have explained the Okazaki fragments, the primer, the primase enzyme, the helicase enzyme. So all these proteins share in the replication process. How many proteins up till now have we used in DNA replication? We have used the helicase enzyme. We have used the DNA polymerase enzyme. We have used the, in addition to helicase and DNA polymerase, we have used the ligase enzyme in order to join the Okazaki fragment. We have used the primase enzyme in order to form the primer. All these four are enzymes, and they are called proteins of replication. And actually, they are enzymes of replication. So why don't we say the enzymes of replication? Because there are still other proteins that are not enzymes mainly, and that help in the process of replication. So we gather them together under one group, and we call them the proteins of replication. There are other proteins that help in replication, like the single strand binding proteins and the topoisomerases. The single strand binding proteins are proteins that bind to the unfolded segments of the DNA molecule to keep them apart, to make sure that the two segments are wide apart enough for the enzymes of replication to be capable of acting. So they act like a scaffold for the process of replication. You're just inserting them in this site to make sure that the bubble, the two template strands on the bubble, will stay distant apart enough to allow the enzymes to work. Similarly, topoisomerase is another protein of replication. So single-stranded binding proteins and topoisomerases are again proteins. So in the previous slide, we mentioned 4 enzymes, and here we mentioned 2. So up till now, we have seen 6 proteins of replication. What do the topoisomerases do? As you can see, the topoisomerases are enzymes, and these enzymes are helping in like making the coils less tight. Sometimes in certain areas of DNA, there are super coils. There is very tight coiling of the DNA molecule. So the topoisomerases, what they do actually is that they make like cuts in the main skeleton of the DNA molecule in order to make the coils less tight, in order to allow the bubble and the template strands in the bubble to stay apart. Okay, so these are proteins of replication. So here you can see the DNA helicase as it is. Of course, the DNA helicase makes the protein, the template strands wide apart. And here we have the topoisomerase that like unfolds areas of DNA that are supercoiled in order to allow helicase to move forward. And we have the single-stranded binding proteins that act as scaffolds to keep the strands apart. So this is another diagram that shows the same concept. So the DNA topoisomerases, as you see here, it makes cuts in the DNA molecule, but they are like transient cuts, meaning that they are temporarily cuts only for the super coils that occur downstream. Downstream means towards the 3' ends of the DNA molecule. So they are not only helpful for DNA replication, but they are also helpful in the process of transcription, as we will see later on, and they mainly work towards the 3' end because the enzymes are moving towards the helicase enzyme is moving towards the 3' end. So they are trying to pave the way for the DNA helicase. So the DNA topoisomerases are usually working at the 3' end, trying to uncoil the supercoils and help the helicase enzyme function. And then we have what we call the DNA proofreading. DNA proofreading is done by DNA polymerase. So this is a second function of the DNA polymerases. This is the second function. The first function of the DNA polymerase was to build up new strands of DNA and therefore help in replication. But you cannot build new strands of DNA and help in replication without making sure that what you did is okay and that there is no mismatch repair. What is a mismatch? There is no mismatch, I'm sorry. So if there is any kind of mismatch, there would be mismatch repair. And this is what we call proofreading. Mismatch repair, meaning correction of any mismatch. And mismatch means that you might have, for example, adenine that instead of having thymine in front of it and making 2 hydrogen bonds might find cytosine in front of it. If adenine finds cytosine in front of it, they wouldn't make a bond. This is first of all, and there would be a mutation, of course, because the specific sequence of nitrogenous base will determine the sequence of on the gene will determine the sequence of amino acids. So it is important to have correct base sequences, because every 3 nitrogenous bases will be translated into a particular amino acid. So if you make any error in this sequence, then you will select a different amino acid, you will have a different polypeptide, and therefore a different function and a mutant protein. So if there is any error, then we should excise or cut and remove the wrong base and replace it by a new nucleotide, a new base in a new nucleotide. So we don't remove the base only, but we remove the whole nucleotide. It's not only DNA polymerase that carries out the repair, but we have 2 other groups of enzymes that are called endonucleases and exonucleases. Now, the endonucleases break in the phosphodiester backbone. They break in the bone, the backbone of the DNA molecule, not that is vertical, not in the nitrogenous bases. And the endo, as their name indicates, if you have a DNA molecule like this. So the exonucleases will break at the edges, but the endonucleases will break the mismatching parts that are present internally, and they break in the backbone itself, and then we replace by the correct segment. But after having removed that part, you insert the correct part. You insert the correct part, but this correct part has to be bonded to the original segment to restore the integrity of the backbone of the DNA molecule, and this is done by means of the ligase enzyme. So if we talk about enzymes of repair, and by the way, enzymes of repair are part of the proteins that control DNA replication. So the DNA replication proteins are many. You will have the DNA polymerase. This is the first one. And then but DNA polymerase does not work alone. So you have the endonucleases. You have the exonucleases. And you have the ligases, just for connecting the segments. So here you can see, this is just to show you the primase enzyme that makes the primer, and every primer is made by a primase, and the primase is also built up in the direction, as you see, 5' to 3'. Any new thing that is built up, any sequence of nucleotides that is built up in the cell is built up in the direction 5' to 3'. And here you can see how the DNA ligase enzymes walk on the DNA molecule to join the newly added nucleotide, meaning that here we had a missing nitrogenous base. So this missing nitrogenous base was inserted, and now it is perfectly in place. So we have a ligase that moves on one strand and another ligase that moves on the other strand in the opposite direction. So if this one moves like this in the direction 3', then this one also moves in the direction 3'. This is only possible because of the anti-parallel nature of the DNA strands. And here you can see, for example, that we had a mismatching because G cannot bind to T. So the G will be excised by an exonuclease, for example. And now we have to get the correct one through the DNA polymerase enzyme, and then we have to use the ligase enzyme. So you excise, and then you add, and then you join. You excise the wrong base that is not mismatching, or that is not perfectly matching. And of course, you don't excise the base only, but you excise the whole nucleotide, and then you substitute or replace, and then you ligate or you join. These are the steps of proofreading. And now we are going to talk about something very interesting that we talked about before, which are the telomeres. We said that every time the cell divides, there are segments from the ends of the chromosomes that are lost, and these are called the telomeres. And these telomeres are non-coding, but as we said, non-coding because they are not translated into a particular protein, so they do not contain genes. They are non-coding, but despite being non-coding, they are highly functional. Why are they functional? They are functional because they act like a clock for the cell, because every cell cycle that is carried out by the cell loses parts of the ends of the chromosomes. And then at a certain stage, so I lose, for example, this telomere, and then I lose that one and that one. And then comes a time when I can no more lose any sequence, because all these are genes, all these are coding. So the cell has to stop. So if the cell lost all these telomeres, any further cell cycle will cause a mutation because it will fragment part of the coding DNA and we will be losing or deleting part of the chromosome, but an important part that is coding. So at that time, the cell has to enter the G note phase of the cell cycle. And not only this, but it would be a permanently paused cell or a permanent arrest of the cell. These protective ends are made of repetitive DNA or repeated DNA. For example, we can have the sequence thymine, thymine, adenine, guanine, guanine, guanine that is repeated hundreds of times. So see here how the telomeres are protective tips. Why are they protective? Because there are enzymes that break down, cytoplasmic enzymes that break down the telomeres with every cell cycle. So why does this occur? Because every cell should have a certain age, okay? And it should die after a certain number of cell cycles. So the breakdown of those telomeres by the enzymes is what controls the maximum number of life cycles that can be done by a cell. So they act as a clock for the cell. Why are they protective? Because if they were not here, the enzymes would hydrolyze or break down parts of the coding DNA. So they are here, they are non-coding because they are protecting the coding. So what do they protect? They protect the genes. So they sacrifice and be hydrolyzed instead of the genes. There is a very important enzyme, or actually a group of enzymes, that is called the telomerase enzyme. And the telomerase enzyme just creates the telomeres. So although the name is called telomerase enzyme, giving the impression that it breaks down the telomeres, yet no, the telomeres are being created, built up, and not only built up, but maintained and replaced by the telomerase enzyme. So this process of losing the ends of the chromosomes or the telomeres is not done without trials from the part of the cell to replace them. So the telomerase enzyme does two important functions to lengthen the age of the cell. The first one is to build up the telomere, and the second one is to try to maintain it. Every time it's broken down, to try to maintain it. But the problem is, if there is a telomerase enzyme, then why is there shortening of the telomeres, actual shortening and proved shortening of the telomeres every time the cell a new cell cycle is added? Because there is very little telomerase enzyme. Otherwise, if the telomerase enzyme was abundant in the cell, no cell would age and no human being would age, and maybe no human being would die. Who knows? So there is a telomerase enzyme, but very little telomerase. And here you can see the telomeres, how long they were at the beginning, and then with every life cycle, the more we divide and the more we get daughter cells. Now we have no telomeres at all. They were long, and then they shorten, and then they shorten more, and then there are 0 telomeres. At that time, when there are 0 telomeres, then the cell cycle or the cell should stop dividing, because any further division of the cell cycle would result in affecting the original genes and resulting in a mutation. Of course, this is extra information just for whoever is curious to know how the telomeres act. Here you have the telomerase enzyme, and then here we have an RNA template, which is the primer. And here there is the shortened DNA. Why is it shortened? Because of cell division. Because of cell division, and during the process of cell division, there are enzymes that break it down. And here are the efforts of the telomerase enzyme to lengthen the DNA that has been shortened. So it brings the bases, the telomerase enzyme, like as you see here, it is again the DNA polymerase that elongates the strand, that elongates the telomere, that repairs the telomere. So what is the function of the telomerase enzyme? It is the site that binds the RNA template on which all this occurs. This is extra information, but it gives you a good idea about the telomerase enzyme that repairs the shortening of the telomere in a trial to lengthen the age of the cell, but unfortunately, it cannot replace it altogether. So the telomeres are an example of repetitive DNA. The telomeres are an example of non-coding DNA, and the telomere are an example of functional non-coding DNA. Functional non-coding DNA. The longer the telomere is, the longer the age of the cell. Now, of course, there is like a general rule that DNA makes RNA and RNA makes protein. So here we have the DNA enzyme and then, the DNA molecule, and then using an RNA polymerase enzyme, we are going to transcribe messenger RNA, and then messenger RNA will undergo a process of processing in order to become mature RNA, and then mature RNA will be delivered to the outside of the cell and reach the ribosome, where it is translated into a polypeptide chain. On the DNA molecule, the strand that is used for transcription of a complementary strand of DNA will be called the antisense strand or the template strand, whereas the other one is called the sense strand. So transcription is building a complementary sequence of messenger RNA on top of the template strand of DNA. Of course, this one is complementary to that one, but if you compare this one to that one, which is the sense strand, you will see that they're almost similar, except for one difference, which is having uracil instead of having thymine. Again, here you have a gene, and that you're going to transcribe that gene. So this is the sequence you're going to transcribe from the gene, because maybe the gene is responsible for making many proteins, but if the gene is responsible for making one protein, then we will transcribe the whole gene. So you will be using the template strand to form a complementary strand of messenger RNA, and this is the process of transcription. And notice that here you have 5' that is complementary to 3'. And then here in the cytosol, this process of transcription occurs in the nucleus, and in the cytosol, the process of translation occurs in the ribosomes, and every 3 successive nitrogenous bases on messenger RNA are translated into an amino acid and are called a codon, whereas every 3 nitrogenous bases on consecutive, and we have to say consecutive, on DNA are called a triplet code, not a codon. So when you say triplet code, you're talking about DNA. When you say codon, you're talking about messenger RNA. I know that we still have the transcription and translation, but because they are important, they will be explained to you on the second part of molecular biology in the second video. Thank you so much. I hope this video was useful. You will be receiving a worksheet in order to like recapitulate what we have said. Thank you so much.